Yes, sir, you're live now.
All right, very good morning, all the students and participants joining for different MOOC courses on Interactomics, mass spectrometry, and bioengineering. Welcome to this second live session. Uh, in this live session, we thought to give you some flavor of latest technologies, which is uh, somewhere relevant to your course curriculum, but otherwise, even as you move forward, you are going to find some of these technologies quite useful in your research. So focus will be on the mass spectrometry-based clinical applications. And in this slide, I just thought to first start with that while we see genomics has really advanced quite a bit over the years, proteomics is still very new. And only in 2014 and 2015, first time the draft human proteome maps were made. And it was only possible because of the advancement in the mass spec. Idea was we know the genome sequences, and earlier we were only trying to predict the protein sequence from the known genome sequence. But then later on, due to advancement in the mass spec, idea was can we look at each organ of human separately? Can we look at the cell lines? And we try to put together all the data to make the draft human proteome map. So that was the first beginning of looking at the whole proteome or the holistic view of proteomics for the human. And that was first study published in 2014 from two independent groups from Bernard Kuster in Germany and Akhilesh Pandey from Johns Hopkins and IOB. Next year, another study from Matthew Sulhan's team in Sweden, they showed the tissue-based proteome map of human, but they use antibody-based approach. Over the time, more and more proteomics data was being generated, and a consortium from HUPO, Human Proteome Organization, led by Dr. Mark Baker, and many teams from the whole world participated in this, including ours from India. Idea was to look at what all human proteome data is available, and can we reanalyze that with a very high stringent manner. So in this light, the first high stringency blueprint of the human proteome map was being made and reported in 2020, and that showed almost 91% of the draft human proteome map, while the initial studies of 2014 showed only 70% or so the proteome map. This is one of the facility which we have at IIT Bombay for the high resolution mass spectrometry based research and training facility, equipped with various type of LC-MS-MS based proteomic technologies. Of course, we have many other proteomic technologies as well, but what you see in the pictures here, we have Orbitrap Fusion, one of the tribrid mass spec technology, interface with the nano-LC. We have the Q-executive mass spec and hybrid technology, and we have the triple quadrupole based TSQ-RTS targeted proteomics and metabolomics technology. So these are various type of tools available for doing the proteomics research. Additionally, we have a startup company, Proteomics International Private Limited, housed at SINE at IIT Bombay, which is catering the only technology and instrument available in India, which is for the 4D omics using the timsTOF, a very powerful new technology, which is giving the proteome data at a very unprecedented scale. So today you'll be very fortunate that you will have the live session and interactions on different type of mass specs, from looking at various type of Orbitraps to moving on to the timsTOF based mass spec technology. So we will be doing various type of demonstrations of these technology today. Now, I'll just give you walk you through briefly what we do in various type of mass spectrometry based proteomics application, irrespective of your sample type, whether you start with the tissue sample, FFP sample, you take the body fluids, cerebrospinal fluid, blood, any kind of samples or saliva. Initially, the goal is to look at the global proteome or the deep proteome analysis of every single protein present in given sample. Or now there is a new emphasis to look at even single cell to avoid the heterogeneity of the disease and cancer. But can we first look at all the protein extracted from those samples, digest those proteins to make the peptides, separate those peptides using liquid chromatography, and then try to analyze those peptides using different workflows of mass spectrometer. There are predominantly two types of mass spec approaches known as DDA data dependent acquisition, or DIA, data independent acquisition. Both of them look at the entire proteome, but just the readout are different. As you can see in DDA, only look at the most abundant precursors in every duty cycle, and then keep doing those fragmentation to generate MSMS. In DIA, we look at the entire MYZ of 300 to 1300 or so MYZ, and look at the fragmentation of different isolation windows across these MYZ. Looking at both the comparison, currently DDA was earlier used for the label-free quantification or the tandem mass tag or iTrag based quantification. Now DIA has become one of the industry standard for doing the quantitative proteomics because you have less missing value, which we see usually in the DDA based approach. Again, I don't want to make it very theoretical and detailed, but just wanted to give you the bird's eye view that to acquire your data and do quantitative proteomics, you can follow one of these two workflow. Currently, DIA is more recommended. Once you have identified the thousands of protein from these samples, you can look at the secondary data analysis, look at which databases search to know, identify the proteins, look at the structural information, do different type of volcano plot or principal component analysis, do interaction network analysis to make a meaningful sense of the data. So this is where something I think, any type of clinical proteomics work which you do, you're going to utilize this workflows. There are many advancements happening in the entire industry. A lot of robotics-based things are coming, different type of chemical labeling-based methods are coming. LC liquid chromatography is very important, and a lot of new type of columns and gradients are being used. And additionally, we have various type of MS instrumentations, the timsTOF technology from Bruker and Astral Orbitrap from the Thermo Fisher. These are currently the most important and the advanced mass spec being used. So again, if the goal was to look at the deep proteome analysis or looking at very comprehensive proteome analysis, we are going to show you the Orbitrap fusion or the timsTOF mass spec to show different type of workflows. You can either label the sample of different conditions, or you can directly do the DIA-based approach, which I just talked to you. But idea is to really get deeper coverage and look at the proteome at a very comprehensive scale so that we don't miss out the proteins to look for quantification. But LCMS is not only used for the proteomics, it can also be utilized for looking at all the metabolites of your samples. It can be looked for the lipidomics, and eventually can we try to combine the data and make it multi-omics. So therefore, other mass specs like the Q-executive mass spec and other orbitraps, which we are going to show you, same sample, maybe you have taken plasma sample or tissue sample, you have prepared the your proteome sample from that, and same sample you have also prepared your metabolome or the lipidome, and now you're going to inject and you are going to add additional layer of information from this technology. This workflows are much simpler and they are going to be taught by TA shortly. Finally, once let's say you have already obtained the list of the proteins and metabolites, which you are very interested as a potential targets or biomarkers, you'd like to validate them. And therefore, a triple quadrupole based mass spec technology, which is the TSQ artist, can be utilized for this purpose. Here, we are not looking at the deep coverage. We are not looking at every single thing. We are looking at only selected ions of peptides or the different other molecules, small molecules. And based on the fragmentation happening from the quadrupole, first quadrupole, we select those abundant interested ions. We fragment those in the collision-induced dissociation cell, and then we further generate MSMS in the second set of quadrupole. So this is known as triple quadrupole, and assay is known as the MRM or multiple reaction monitoring. Similar type of work would also be done on the Orbitrap, which is known as the PRM or parallel reaction monitoring. So this is what broadly I just thought to give you the heads up. We are going to give you various type of live session of different instrument and technologies from the lab directly today. And I'll now introduce you to Dr. Argya. He can turn on his camera. So Argya is the, of course, he's finished his PhD from IIT Bombay, and then he started the entrepreneurship journey as the director of Proteomics International Private Limited, where we are hosting this at the Sign at IIT Bombay, this company. And now he's also TA for this MOOC course. where he's going to give you the workflows for different type of sample prep and mass spec instrumentation. So over to you, Arya. You can give the brief plan and then followed by the instrument demonstrations.
So welcome everyone.
You're audible, just make it full screen.
So welcome everyone. So I will be initially giving you the brief of how the samples are prepared in the mass spec for the proteomics experiment. And after that, we will work through the real-time applications, how the samples are loaded, how the instrument works into the basic crisp view of that. So initially, you can take your clinical samples, either blood. So here, let me go to the pointer option. So what you can find, like you can initiate the proteomic samples either with cell or tissue, or you can go with the blood samples also. So once these blood samples have been taken or the cell or tissue samples have been taken, the initial processing happens. For cell and tissue samples, we go for the cell lysate, cell or tissue lysate preparation in the presence of the buffer. So selecting the most appropriate buffer is very important because your proteins will be isolated on the basis of the buffers you use. Initially, you can use an chaotropic agent such as urea or an urea lysate buffer, which is universally accepted. And you can use little bit amount of detergent also in that cell or tissue lysate buffer. But keep in mind, like if you are using an detergent, you have to clean that detergent before the samples are being injected. So once the cell and tissue lysates are prepared either by mechanical bead beating or by shearing action, you go for protein quantification. The protein quantification happens in the plate reader. You can use Bradford or BCA, which are routinely used for the protein quantification detection, followed by the digestion of the peptides. In the digestion, you have three major steps. One is reduction, one is alkylation, and further is trypsinization. Once that happens, you do a cleanup, you do the cleanup of the peptide. Once the peptides are getting cleaned up, you can see like this is the timsTOF Pro with the Evosep Nano LC. Generally, with the routine HPLC, you have to clean up the samples before, like you have to remove the salts before the samples are injected into the mass spec. But here with the robust Evosep Nano LC, you can directly load your samples into the Evosep tips. And once those are procured, you can go for data visualization. Just coming to the steps of protein extraction. So we generally in our lab, we use 8 molar urea with 50 millimolar concentration with of TAB and NaCl. The TAB maintains the osmotic pressure as well as the NaCl means facilitates maintains the stability of the buffer. Always, each time when the lysis is being done, always use the fresh buffer. Once you take the samples in this, once you take your samples, you go for the lysis. You can use mechanical shearing such as sonication or the bead beater where magnetic shearing happens. Once your protein lysate is being taken up, you do quantify the samples. You run the SDS page to see your gel patterns, and once the gel pattern has been done, you can excise the gels for the in solution for the in gel digestion, or you can directly proceed with the lysate, which is in solution digestion. So always estimate the conduct. So the point of caution is always use means estimate the standard curve. The accurate protein confirmation is required. Bradford can be performed, but if you have some detergent which interfere with the Bradford activity, you can use BCA for that. So this is a representative of the Bradford curve. So what you are seeing here, and once those are done, we will proceed with the first step. After a digestion, we will proceed with the first step, which is called reduction, where 20 millimolar TCEP is added, tricholine phosphine. Once that is being used, we there is a chance we incubate it for 1 hour, and after incubation, there is a chance that uh, the peptides which have been digested means the proteins which have opened up due to the reduction of the TCEP might refold. So we add indole acetamide like the IAA, for alkylation so that the protein does not refolds it again. And once the protein is now open, we go for protein and Bradford digestion. So now we will move on to the sample demo. So what you can find, like we have already measured the tissue samples into the Appendrop, like 50 mg of tissue samples will be great to start with. Once you use the tissue samples, you take the Appendrop, skip it in the ice. So
Yeah,
once it,
just one second.
yes.
So we can highlight, this window which you are showing for live demonstration.
So this is the sonicator which is generally used. So this is the sonicator which is generally used. You can place your samples inside the sonicator. There is this probe which produces the sonication 5 second on and 5 second off. You can sonicate your samples inside by dipping this inside the tube, and at that 4 degree is maintained with the ice, because this kind of sonication creates lot of heat. So which can be used for sonication. So this is the mechanical bead beater. So this is the bead beater. So what you can see, you can use this instrument also for the sonication. So you just place your samples here. So you just place your samples inside the magnetic beads. Like this is the tray. You put your samples inside, put the magnetic beads inside, put the sample equipment inside, and you place this apparatus inside. Now you can set the temperature, you can set the time here. If you want to go for 15 minutes sonication, you just increase the same. If you want to go for 10 minutes, you can reduce the same. So and the modes can be standard, low, and high. Some, if the tissues are very hard and very brittle, you can choose high standard sonication. And if your tissue is forming very froth, like froth kind of things, you can go for low standard sonication, okay, in the low mode. So whatever you select, so suppose I have selected the low mode for say 9 minutes, and you just start. So it has already logged, and you can see my timer has started. It has already initiated the uh, 9 beats, and the, after 9 minutes, my samples will be coming up. So as this is being the demo, I will take out the sample. This is the apparatus inside. Very well. So now my sample is lysed. Now we will initiate with the Bradford quantification. Sorry, SDS page gel. So this is the gel apparatus what we are having. So this is the SDS gel apparatus what we standardly have. We put the gels inside. So this is a gel piece which is placed inside. You just take out the comb, and we can load 10 microgram of the proteins. So you can go for replicates or triplicates or duplicates, like to show how the gel comes looks like. So this you can find, like this is the perfect blend of gel which comes. So once you see the protein forms nice band or the pattern, like nice bands inside the gel, you can then further go for the in solution digestion. So for the in solution digestion, you take the samples, like your once your gel is good to go. So keep everything and do it in the ice. So you can determine the Bradford quantification, means after the lysate is being prepared. This step, next step is the Bradford preparation. So we have already prepared a gel, and based on the colorimetric assays, the Bradford is being determined. So Khushi will, my TA Khushi will tell out about the Bradford assay.
So we use this data and this is a graph, and for this the R value is 0.99. And
So once the Bradford is being done, we will next initiate with the sample digestion. So we will first initiate with the alkylation reduction process. So you can take the concentration. So you put the alkylating buffer into the lysate. Sorry, yeah. You put the reduction like the TCP in the lysate. Once the TCP is been done, you store it at 37 degree. You can use the heating block, or we have an incubator which is at 30 degree. So once the incubation is then being done, once the incubation is completed, bring out the samples. We will add IAA. We'll add the indole acetamide. Please use pen drops like the amber color pen drop bottle. You incubate it, you put the samples inside, and once that is being done, once that is being completed, you again incubate it for half an hour, like 1 hour incubation for the reduction step and half an hour incubation for the alkylation step. Once your alkylation step is completed, you can put the trypsin in that. So 1:20 or 1:30 ratios of trypsin can be put inside. So you put your trypsin inside in the concentration ratio of 1:30 or 1:20, which is widely accepted. And we incubate it for overnight for 8 hours. Now, two kinds of methods can be optimized. At 37 degree for 16 hours or 8 hours, okay, we consider that total digestion is already been done. Okay, so we consider that the digestion is already been done. But if you can do another second step which is called as double digestion. Okay, you can perform another step of double digestion for that. So let me share the slides again. So you have seen the sample preparation demo. So once your peptides, you can perform a double digestion. That means add 4 microliter or 1:10 concentration of trypsin again, so that the digestion efficiency increases. You next perform the peptide quantification, in which the peptides are reconstituted in 20 microliter of this buffer, like 0.1% Fa or protonating agents or anything which you consider. This is a plate reader, and with the same thermomicrodraft plate, you go for this quantification of the peptides. One year quantification of the peptides is good. We inject generally 3 nanogram to 1 microgram of peptides. So we then move on to the mass spectrometry. So now we will go for the live demonstration of this.
First, we are starting with the timsTOF. Okay, all right. So sample prep could be very tricky, and it can sometime, you have to really understand your biochemistry very well, because when you will see different type of sample, you have to try different ways of lysis or different type of solubilization. There cannot be just one single method of doing the sample preparation. But what we just showed you, like some, the flavor of the general step involved of how to make lysate, how to quantify that, how to look at qualitative changes and look at the GSP profile. Those are very basic but very important. And then you are going to digest that protein and get sufficient peptides. Those peptides are to be cleaned for desorting to ensure there is no salt. And then you are going to quantify that peptide again. And eventually you are going to inject that peptide in mass spec. So what we are showing you right now is one of the instrument orbital fusion, where around 1 microgram peptide will be injected. And Arge is going to take forward now the demonstration of that.
So welcome. So this is the current orbital mass spectrometer, which is orbital fusion, and this orbital fusion is connected with the 12,000 nano LC. So this is how the nano LC looks like. It has the quaternary pump inside. We use the bisolvent, like the one is the hydrophilic, which is 0.1% Fa, and one is the hydrophobic, which is 80% of ACS. So this is the sample injector here. So you can find like 96, like at a stretch, you can usually go on to 28 slides, and these are the vials in which the samples are poured. Like what Prof explained, after your sample is reconstituted, we put around 20 microliter of the samples inside. Once the samples has been placed down, once the samples have been placed up, we put it inside. It has various other functions, but this is one of the accurate instrument or the nano LC for the proteomics workflow. So once the sample has been injected, so once the sample has been injected from the nano LC, it is diluted, it comes to the trap column. In the trap column, which is a pre column, if there is a certain abnormalities, the samples they may get cleaned there. Otherwise, it will be going to the main column. So this is the our elution column or the step column or the nanospray column, what we are using, what you can see. So this column generally is a C18 column. Now on here, actual sample preparation happens based on their hydrophobicity and hydrophilicity, whatever gradient we apply. The sample then goes inside. What you can see is the ITC. Here the space route ionization happens. And once from here, it generally goes inside the system. Now, what we are showing, so what we are showing is generally one is a 2 hours gradient, and you can find like this is how the separation of the chromatogram happens. I will request Suresh to show the MS and MSMS peak. So here you can find we have just expanded the chromatogram, and these the illusions are the MS, and between two MS is the MSMS. Generally for one MS, at least you consider 20 MSMS fragments. And if it is 20 MSMS fragments, the PSMs comes good in the discovery. Actually your data is very good. So this is how we generate the quality of the data, or chromatogram of the data is coming good only. Okay. So once this 120 minutes experiment is done, okay. So now you have the global data, and then you can correlate. Then you can basically correlate the same. So once that is being done, you can basically correlate the same with the FASTA, and you can do the you can do a FASTA or an web search apparatus, and you can find. I will now move on. Like with the same samples, you have done the proteomics experiment. There is another mode which is called metabolomics and lipidomics. Just to give you a brief, these two profiling can also help to identify the markers which can be very place and critical role in clinical applications and disease detection. So I will point out you to the instrument. So as you can find, like this is the instrument in which we perform metabolomics and lipidomics. We have an HPLC 3000 series HPLC which is connected. with the orbiter Q exactly mass spectrometer. Now I request Amit to give the.
So as you have learned about the proteomics, so there you can there you have learned that you need to chop the proteins to peptides, but it is not the similar case for metabolomics and lipidomics. It's very straightforward. With the help of only chill methanol and Prof, you can extract the entire lipidome and metabolome from the serum or even the two samples, so the similar samples that you have used for the proteomics. So now the machine you are seeing here, these are actually the QE mass spec. So it has two different mass analyzer, quarter color and orbital, and there are two different setups. One is the HPLC and one is the mass spec. So in the HPLC, you can see three different compartments there. The very first compartment is the column. So this is the column compartment here. So here you can see one of the key component of this entire system is the column here. So this is called hyperseed gold column. So this we use exclusively for metabolites. Yes, so sometimes you also even use lipidomics here. So this is another column. So it is called CH8C18 column. So every column has their own different chemistry and own different particle size to differentiate among those small molecules. Now on another compartment is the auto sampler. So in auto sampler, you basically need to put the samples here. So this is the auto sampler. That in one way you can keep 120 samples here. So these are the different vials, the transparent vials you can see. So here there's another column called insert. So in insert you need to put this kind of your samples for the further acquisition. After this, you will also have your pump here. So pump, you basically try to monitor what is the flow rate and what is the pressure of the entire system. And this is the solvent system here. Like for metabolomics, we use different kind of solvents. Also for lipidomics, we try to have a quadrant solvent system. Now here you can see a quantum of global proteomics here, global metabolomics here. So you can see multiple peaks are here. So that is going to give us the information about all the available metabolites that are coming from the serum or even the samples. So metabolomics can have lot of variations. That's why we try to use a reserve bind, which is a non-endogenous component that is not found to be in any kind of sample. So basically, try to spiking the reservoir, and you try to see the RT of the reservoir is consistent or not. So this is the time series plot. You can see the plot. The tolerance is under plus minus 2%. So this is the QC of the global metabolomics. Now, one of the another important things, you can also go for targeted metabolomics here. So the difference is that so difference is that from the global metabolomics, you can have the entire the bird's eye view of that what are the metabolites are getting detected. Now you have some cohorts of the different region of India. Now suppose you want to validate those metabolites in their different parts in a different cohort. So for that so that the global metabolites give you the information about how the metabolites are getting detected, and you get the MS2 fragmentation, and you will give the information to the here what are the different fragments of the metabolites. You can see the model of the targeted metabolites, where you can see specific fragments of your interest of metabolite will be coming in the model. So now one short demonstration with the mass analyzer. So this is the from here the solvents and the solution and samples will come, and in the inside you will see a sweep cone here. So basically there is another component for ITC. So inside there will be a temperature 320 degrees Celsius, and with that extreme heat the metabolites are going to get fragmented, and then they will go to the subsequent mass analyzer, the orbitrap and the quadrupole. So then from there we will retrieve the data and we try to analyze with different type of commercial softwares.
So once you get the metabolomic profiling, you know what all metabolites are there. Using the metabo analyst software for analyzing the data analysis protocol, and you can use various searches such as lipid search for lipidomics, and compound discoverer for the metabolomics for tabulation. This is another instrument which, to be briefed, what professor told, we can perform SRM and PRM on this instrument. This is a triple quadrupole all this, we have a higher version of the HPLC here, which is Vanquish HPLC. Similarly, you place your samples inside the component. So this is the sample compartment. You place your samples inside. So this is the sample compartment. Once you place your samples inside with the pumps, like the samples are getting eluted. From the sample elution, it directly comes to the column compartment. So this is the column compartment, and once the samples are inside this, like once the samples get inside, it directly comes to the heavy source where the samples is like where the samples are now being eluted. And once the sample is getting eluted, you can find the SRM and PRM based applications, which can routinely use for SRM and PRM based applications. So back to you, Prof, for the introduction of the timsTOF score.
All right. So what you saw, like, mass spectrometry, while it looks like one instrument or one technology, but there are different type of specialized mass specs which can do different type of application. And you just saw three type of mass specs for looking at the global proteome analysis, looking at the global metabolome or lipidome analysis, and target proteomic analysis. Now, even global proteomics can be done on different type of instrument platforms. One, what we showed you was the Orbitrap technology. Now, what we are going to show you is the timsTOF technology, which will be demonstrated by Dr. Shambhu. And this is one of the very latest technology, which is based on the 4D omics. Traditionally, we have been looking at retention time, looking at M by Z and the intensity, but this particular technology also adds up the cross-sectional area, the looking at spatial separation as a fourth dimension separation. And with all the new innovation, it is now able to give us the depth of the proteome coverage, which was not seen earlier. So this is an instrument which will be shown shortly, but just to give you a flavor, traditionally what we used to get almost 3,500 to 4,000 proteins from the complex tissue or the cell line samples. But using this technology, we are easily able to reach 8,500 to 9,000 proteins from individual samples and crossing over 10,500 proteins from the protein group analysis. So that's actually very paradigm shift to almost 2 to 3 fold change in the proteome coverage, which we can see, right? So Shambhu will show you now the timsTOF Pro 2 and the EvoSep chromatography technology.
Okay, thank you, sir. So this is the timsTOF Pro instrument, and it is integrated with the Nano LC, which is from the EvoSep one. So here you can see for mobile phase in this system, we use 0.1% FA water, and which is hydrophilic in nature. And for hydrophobic, we use 0.1% FA in SEM. So this is the, and this to make the mobile phase for this. And here, this is the Evo tapes where we load our samples. And when we start the run, so this robotic arm pick up this Evo tape and will inject the samples here. And from this, peptides will come through this transfer line and it will reach to the C18 column. So in C18 column, peptides will be separated based on the hydrophobicity of the peptides. So after separation based on the hydrophobicity, peptides will go inside the ionization source where ionization will happen. And then ions will migrate to the TIMS where ions will be separated based on the ion mobility, and which is decided by the shape and size of the ions and charge of the ions. So, and next, after the ion mobility separation, it goes to the passive where fabulous accumulation and serial fragmentation occurs. And then it reaches to the top time of flight where M by G ions are detected. So all these components are handled by the two software, which is Bruker Compass High Star, where we line up the samples, where we place the position of the vials of the samples. And another Another software is the Teams Control, which control the system for the calibration things. So here you can see the previously run data. Here you can see the chromatogram, how chromatograms looks like. And this system is also integrated with the uh, Bruker proteoscape search, where you can also identify the proteins during the runtime. So these are the previous run, which you can see. And we can also, in this system, we can also perform the group-wise analysis. So group-wise analysis look like this. Okay, so that's all from my side. Over to Professor Sanjeeva.
All right. Yeah, so what participants can appreciate that uh, the new advancements, especially the timsTOF as one of the technology, and then we have the Astral Orbitrap. With these advent of these new technologies, proteomics has now reached to a scale which was very unprecedented, which has never been seen. So now one could relook at many of the clinical problems and try to see, can you identify the right biomarker and right targets from the complex samples. So now Arga is going to show you different applications which are possible by using these instrumentation and these technologies. And again, LCMS is something which you are going to use it for different type of application, and that you are going to get some brief overview on that. So Arga, you can now talk about some of the applications.
So welcome everyone. So just to carry on, like we have given you a taste how the peptide quantification is being done. You have gone through the demonstration where the peptides graph have been like once the concentration of the peptides have been needed, and then the same peptide samples then further can be used for either Orbitrap based quantification or the SRM or PRM based application for targeted proteomics, or you can perform a more robust DIF assay using the timsTOF Pro. So we have shown you the Orbiter fusion demonstration. We have given you a metabolomics demonstration using Q Exactive. And finally, we have also given you the timsTOF Pro 2 demonstration. Now coming to the therapeutic drug monitoring clinical applications. Now these technologies which are now currently being used in the application, what is their real life applications, like where they are used? So basically in the therapeutic drug monitoring, these technologies are means very importantly used in the fields of detecting the immunosuppressants, the antibiotics, the thiopurines, and they play a crucial role in the pediatrics also. The important immunosuppressants such as tacromylus, cyclosporin, the cyromylus, which helps for means basically during the grafting or the organ transfer, these antibiotics or the immunosuppressants plays a very crucial role to means the most to play a most crucial role for the uh, recognizing the self, means whenever a foreign organ is donated in your body, like whenever an organ transplant is done, that is a foreign to the body. So now what happens if that organ is identified as non-self? Many antibodies and the cytotoxic T cells are being generated, and due to which there is a damage, like there is a damage or a rejection of that organ. To depress that, to minimize that, the immunosuppressants such as tacromylase, cyclosporin, or during the grafting of the skin, this kind of immunosuppressant is used. And based on the concentration of the immunosuppressant, when do they go for the drug or the metabolites have been used for the cross reactivity assay. So as you can find, the tacromylase, it is routinely used for the tacromylase, and 5 to 50 nanogram per ml is easily detected to prevent the cross reactivity. 20 to 30% of reduction in the acute rejection have been done, means versus the immunoassay monitoring. And after this drug has been successfully monitored with the optimized, the tacromylase drug has been, like tacromylase drug has been successfully monitored, the reduction of the kidney or the toxicity have reduced from 25 to 40%. Similarly, the vancomycin, the aminoglycosides, around 10 microliter of the dried blood spots can be used for the detection of these antibodies in the neonates. Nephrotoxicity or the organ failure or any initial diseases which is to be screened in the neonates can be easily identified with the help of this antibody concentration. Plotting the area under the curve and minimal means MIC optimization can easily help you. Thiopurines, the 6-TGN, the 6-MMP, and the IBDs are basically used. And when the TGN ratio, like once you determine the concentration of the TGN to the MMP ratio, when it is less than like more less than 20 is to 1, means it is like quite safe. The remission rates are around 45 to 68%. Okay, and toxicity prediction can be easily done. In the in the pediatrics, batricininib, the JAK inhibitor, 25 microliter of the plasma is generally used to enable the PK pharmacokinetic studies in the children. First time the pediatrician in the mass spec was used to observe the means therapeutic drug monitoring was personal. So these can also be channelized as the personal doing. Not only that, in the newborn screening, like how the mass spec plays a crucial role in the newborn screening, the amino acid disorders such as PPKU, the phenylketonuria, the homocystinuria, the tyrosinemia, which is in the newborns or the means neonates, can be used. Basically, the amino acids such as phenylalanine, leucine, isoleucine, valine, and methionine are basically detected where the tyrosine is being detected. The phenylalanine to the tyrosine ratio, if it is more than 2.5, specific elevations are happened. So this is the diagnostic strategy, and this is how the clinician's intervention plays a role with an recommending a food with dietary restriction and enzyme replacement can be happen. Organic acidemias, the methyl malonide, propanoid, and glutaric type 1 plays an important crucial roles in the organic acidemias. Generally, the propanoic acid, the glutaric acid, and their ratios, the C3 to C2 ratios has been drawn out with the identification of their concentration with the help of HPLC and the mass spec, and the pattern recognitions or ratios have been identified. Clinical interventions can help us to understand like the carnitines, the vitamins, and the vitamin B12 dialysis if required. Okay, in these patients having organic acidemias. Fatty acid oxidation, the branch change lipids, the branch change and fatty acids such as MCAD, VLCAD, LSCAD, the C8, the C14, the essential fatty acids, like you take out the ratio of the C8 to C10s using the acyl carnitine profilings. Okay, and once you determine the acyl carnitine profiling, the glucose type 4 medium chain triacids can be used for the clinical intervention to identify and means fix this fatty acid oxidation charges. Similarly, urea cycle disorders such as citrullinemia, excess of citrulline in the blood samples, arginosuccinate aciduria can be easily detected with the citrulline and arginosuccinate metabolites. The diagnostic strategies they generally used to predict the low citrulline and the high arginosuccinate concentration, and ammonia scavengers and dialysis can be performed. So this is one of the latest things which are happening in the newborn techniques, like the screening of the newborn. The technical specs are around single 3 DBS punch can be taken, dried blood spot, like whenever means basically you cannot draw a whole blood into the vial. So basically, one drop of dry blood is taken into the slides, or it can be it can be dried into the filter paper, like the filter paper, and 3 only just 3 area of that dried blood spot punch is being taken, which can detect 60 metabolites. Like these are the very much targeted, they have detected much more, but the panel of 60 metabolites, like including all these detected metabolites, can be used in just a 2-minute run. So whatever you is shown, we have shown TSQ, altis, triple, quadruple, that can be used for just a 2-minute run to determine the false negative and false positive for all these diseases. Coming to the metabolite profiling mechanism, most of the concepts are the same, but in the analytical style, multi-metabolite quantification, diagnostic ratios, pattern recognition, and second tier testing can be performed. In the multi-metabolite quantification, The technical processes of 11 amino acid and 31 acyl carnitines are being used. They are absolute. They are basically done for the absolute quantification across the dynamic range, and all the 50 plus disorders which is happening due to the metabolic dysregularities or metabolomic reprogramming can be detected. Diagnostic ratios, the C3, the C2, C8, C10 phenyl tyrosine ratios can increase the specificity and reduce the false positive. Like the MCAT can be easily detected if it is more than 0.44 of ratio. The PKU, ketonuria, if the phenyl to the tyrosine ratio is more than 2.5, then the person might have the chance of having phenylketonuria, which can be easily. Pattern recognition molecules, multiple abnormal markers create signatures, which can be used as for the confirmatory diagnosis, such as glutaric acid, the C5DC. When it shoots up, it can result into the understanding of that. Similarly, C3. The second tier testing, the GCMS, like here another technique can be used, gas chromatography. LC can also be done, liquid chromatography can also be done, which can be a definitive diagnosis confirming the borderline results of MSMS. Now, coming to the most important, there will be a multiple research on the cancer biomarker discovery using the MSMS, but we have tried to focus on very few which are routinely being practiced or at the advanced stage of FDA approval. So, coming like each cancer biomarker discovery has at least three phases if you are performing like what professor explained. If you are performing a biomarker discovery, first in the discovery phase you take the label free LCMS data, like the from the cohort of patient samples, like 50 samples or 100 samples. You take the cohort of data, and whatever data you get, you do increase the coverage. You can identify the more numbers of proteins, you can do in-depth analysis to increase the protein coverage, and around 500 to 10K tumor tissue or plasma or proteins which can be secreted into the biological fluids are identified. The very significant proteins are then further taken, like 50 to 200 proteins, which we generally do a discovery proteomics study. Around 50 to 200 proteins are further taken for the biomarker candidate result, and 10 to the power 12 dynamic range and high abundance masking has been discovered. Like the proteins which are very much robust across the cohort are taken further. Then you can plan a verification, which professor have already explained. Like we can perform the multiple reaction monitoring or the parallel reaction monitoring. Now this is very important. The clinical cohorts, you do a blinded study on 100 to 500 patient samples. That 10 to 20 samples which you got from 5,000 to 10,000 proteins, which have been filtered now to 10 to 20 validated markers, you is further taken. Their reproducibility, their matrix effect, their specificity, their selectivity is being observed. And once they all process the criteria and the CV is less than 30% or 20% to the stringent case, it is further taken for the technical part. In the technical part, immuno-MRM is being performed where antibodies are enriched for that particular tissue samples, and we spike it for the sensitivity using plasma, and clinical gate assays are being developed for or the antibody development. This can go for the antibody development. So this is the cancer biomarker example. For the prostate, multi-marker panel is being used where 7 proteins are being observed, and around the AUC is around 8.89, and the PSA versus the PSA prostate cancer is around 6.68. It is under the status of clinical validation. In the pancreatic markers, the CA19 has 9 proteins which are cancer embryogenic antigens, as well as 3 novel glycoproteins is being used. The sensitivity currently reported is around 92 to 95% of sensitivity. The phase 3 trials for these pancreatic cancer markers have been initiated. And for the lung cancer, EGR phosphopeptides are quite well-known markers, like the quite well-known proteins which have been dysregulated. This predicts the TKI, and it is under research use. Coming to the multiple myeloma application, coming to the multiple myeloma applications, in the multiple myeloma diagnosis, their monitoring, their isotyping, and their workflow is being used. So generally, this is the mass fix test which is generated by the Mayo Clinic, in which the multi-top is being intact, means used for the intact IgG or you can quantify the M proteins also. The isotyping can be used by our instrument also, like LC-MS, MS status, and the whole things can be automated within 10 minutes using the LC. So this is the LC-MS. So this is the power of the LC-MS, like a test which goes for 1 day can be easily robusted, and 10 minutes it can be automated. When you are performing the diagnosis, the sensitivity of the test using the multi-top is around 95.24%. Similarly, for 100% diagnosis, these tests can also be optimized with the orbitrap-based mass spec as well as the timsTOF-based mass spec. They are complementary to the conventional techniques of IFE, like isoelectric focusing electrophoresis, the SPEP electrophoresis, and basically the light chains of the myelomas are only detected, like in the case of the diagnosis, once you detect the light chains. But when you do the minimal residual monitoring using multiforms, around 0.05 gram per liter of the LOD can be tracked down, which is 10x better than the gel technique, which is the IFE. It tracks down the treatment response and also presents the early response for the detection. You can detect the IgA kappa and IgA lambda antibodies, IgG kappa and IgA kappa antibodies. That is definitely for isotype ID, and this is the treatment selection for which kind of multiple myeloma, like in which kind of multiple myeloma, what therapeutic interventions can be done. So once the 10 minutes automated analysis is being set for all these method, no reagent is required, whereas the gel techniques, traditionally gel techniques requires 10 to 2 to 3 hours for the operation only. Further means analysis of them will take one more, like 5 to 6 hours goes in the first line of screening when you are using gel based. So other important technique other than multiple myeloma is like you can detect all hemoglobinopathies using the LCMS. So few of them are listed here. You can use to detect the sickle cell anemia, beta thalassemia, the HBC, and HPFP. So in the sickle cell anemia, at the beta subunit, at the sixth position, glutamine is being replaced by the valine. There is a 30 kilodalton mass shift, and you can usually understand if the person is having a HBSS or not. So this can be used for the penicillin prophylaxis, like the clinical actions can be taken, and this detection of the sickle cell anemia has bring down the mortality less than 80%. Similarly, for thalassemia, you can see the down regulation of beta globulin down expression and up expression of alpha beta with a ratio of 2. This can help in the transfusion and chelation, like transfusion of the fresh cells, fresh blood into the paper, and it can also help in the prenatal detection. Similarly, for which are the structural variants of hemoglobins, the counseling and the monitoring can be performed, understanding like which can be the carrier hemoglobin molecule. Similarly, for HFPF, the gamma chains can be like the fetal hemoglobin is being monitored, and this can be prognosis for SAD. Final but not the least, the personalized medicine framework can be done with the help of these mass specs. Like lot of clinical wellness studies have been done in today's world with this modern technology. So you have to just understand in the personalized medicine there is 4P, which can help into the detection of the longitudinal wellness or the patients which might be prone to the diseases. First is the predictive state line, preventive, personalization, and participation. What is the contribution of the mass spec into it? You can detect the baseline proteomes. It can further in the preventive after understanding which proteins are dysregulated or which metabolites are dysregulated. You can perform a pharmacokinetic proteomics, like which drugs work best for this kind of diseases.
So,
Which
I think this is fantastic, but I think we have to stop here. So, as you can see, there are many applications possible using the LCMS technologies, irrespective of, your goals of looking at which course you are working on, interactomics, bioengineering, or mass spec. You will find these applications very relevant and very useful. And that's where knowing the some fundamentals of mass spec and different instrument configurations are going to be quite useful for your entire career. So I hope some of these demonstration which we showed you in the live session, additionally we have in our videos many lectures covered, will give you a good flavor of it. but the way the technologies are very rapidly advancing in this area, and so are the different softwares and data analysis workflows, it's a good idea for you to, to follow the latest literature and the published articles. Also, many open access good softwares are available. Some of the software which our lab has also contributed. Try to start going through them so that you are getting more and more familiar. And please feel free to ask us more questions in the forum or the live sessions, as well as, in the different Telegram groups which we have made. So we wish you all good luck for, continuing this MOOC program. And we'll again be having one more live session in the next month. All right. That's all from our side. Let me thank Dr. Argya and all the TAs for doing the demonstration today.